They're getting random data.
Okay, let's have a class.
There's already statements.
So, first of all, I would ask you guys some of the questions what you did for your validation test. AEM test. Okay, which one is Indo positive? Which one is Indo positive?
E. coli.
Okay, good. E. coli is Indo positive. So always remember, this is a very easy test for Indo. Okay, quick test for E. coli is Indo test. It's positive. And always remember the reagent we need to add is Kovac. Kovac on the top, and it will show you the color is cherry red. So I use red color, a red chalk pen to draw it. Okay. Okay, phenylalanine deaminase test. Phenylalanine deaminase test. And remember, we need to add ferric chloride. It shows you dark green color. Is positive. Which one is positive? Which one is positive? Based on what you did. Confirmation test. Then, which one is positive?
Citrobacter?
Yeah, citrobacter is positive. Very good. Citrobacter is positive. Okay, and we said this is a surrogate for Proteus vulgaris. So very good. Okay, these two are important previous review the results, and you should record it on your notebook. Okay, today's work. First of all, we need to do confirmation test. Okay, how to do confirmation test? Oxidase test. You should have agar plates for oxidase test. And it should grow. Is that correct? Should grow very well. Okay, what you put there? Did you put citrobacter on one side? Yeah, that's citrobacter. And the other side, I said, any bacteria you like. Let's say some of you put E. coli there. And they should grow. We will add is called oxidase reagent. So we're going to add there oxidase reagent. And I also want to tell you one thing. Please do this later on, because the plates we're going to use for to do enter tube. It's a beautiful pencil. We'll show you. So do that later. We have oxidase reagent is right here. I have a pipette prepared to you. When you go inside, absorb a little bit, tiny little bit. Don't have to be 100 microliter, okay? Tiny little bit, even like 5 microliter is good enough. Tiny little bit. Add onto the smear there to see which one showing you purple color. The side showing you purple color is positive. So this test is right here for you. You can come to the front to do after you do the enter tube. Okay, enter tube is right here. So once you dump oxidase reagent, when you see purple color, it is positive. I know this color you may not see it, but purple color is positive. So that's confirmation test right here for you to do. Do this later on. Okay, that's confirmation test number 1. Number 2, MRVP test. Okay, you need to have an empty tube. So you will get an empty tube right here. Everybody need to get an empty tube. Okay, so how you do it? This is your empty tube. And this will be your solution. You're already growing there. Doesn't matter, it's either E. coli or Enterobacter. Okay, so how you do it? Dump about half solution here. About half solution here. And then mark this, label this as MR. And the original one as VP. And then you need to come here to the front desk to get methyl red reagent. And then have 2 drops. And shake very well. So methyl red reagent is right there. This for methyl red test positive. If the color is changed, it's positive. If no change, it's negative. Okay, how about the MRVP test? The original tube, we need to add VP reagent 1 and 2, or we say A and B. Let them react it. Might be need to be a couple of hours. So you could put this tube in back to your rack and do the observation on Thursday. That need to take a couple of hours. Okay, so methyl red, you could be just add 2 drops of methyl red reagent and then do the observation immediately. VP, we need to test in the left solution in the tube. We add VP reagent A and the VP reagent B is right here. This is the VP reagent B, and this is the VP reagent A. And there is a couple of here, you can grab it and return to the front desk. And you drop couple of drops. Each one, I would say 2 to 3 drops. 2 to 3 drops of each. And then will have to be storage. 5 hours, so just left this in your rack and do the observation on Thursday. Is there any color change? If I believe if one of them is pink color, it is positive, otherwise it's negative. So we'll see it, okay? Okay, today's lab, we will do a couple of things. Number 1, enteral tube. So enteral tube is a multi-channel biochemistry test. Looks very beautiful, like a pencil. Like a pencil like this. What's the idea of this? We doing all these biochemistry tests is one by one reagent by reagent. It's tedious, lots of work. So we have those tools, which is really nice, have 12 biochemistry tests, could do at a time. So you see these wells, every well is containing different reagents. And if you read, they label them as glucose, lysine, ornithine, sulfide, indole, adenosine, lactose, albinol, sorbitol, VP, phenylalanine, urea, and citrate. So which means all these substrates already been in this well. And then you have these needles. These needles can pick the colony from the agar plates and going through all these well and come back. Then we close these 2 caps and do the incubation, and we will see the color change after the incubation. And then each color change, positive or negative, we have to compare to the standard code book and write down the code. And if the code is exactly following the code box, then we will identify that bacteria. Now, here is something I want to do is I would love you to practice how to do this, but we're not going to read the code. Okay, we're not gonna read the code book. And after this, incubation on Thursday, as long as you do not open the cap, it will be a very beautiful color pencil. So you can take it to the home as a gift of this class. Okay, so I will be showing you, doing an example to you how to do right now, and you can do by yourself. It's just for fun. So how to do this? Let's wear the gloves first. That's the first thing, as usual. Then you guys have to do is a label. There is a side here called name. You have to label it. Okay, label your name, batch number, and what type of the bacteria you put it. That's fine, and you can add it. So let's borrow one plate from here. I'll just borrow one plate. This have Citrobacter and E. coli. I would do E. coli. Okay, how to do it? We gonna be taking off this cap. Let's look at this. Okay, black, a white one, it's a needle. I'm just doing observation for you. The blue one is a kind of like a clicked holder. It's like a holder. Okay, so how we do it? We will do is that I will take it off from this side first. Okay, hold on the hands, and this side I also gonna hold on the hands. So I will be holding like this, and I will pick from the E. coli side. So the E. coli side, I will pick some colony, just some smear. And then what I do? I'm gonna going through the whole well, very gentle, very gentle, very gentle, very gentle. Don't take it out. If you take it out, you can't put it back, because it's contaminated with bacteria in the air. So then very, very gentle, very gentle, very gentle, put it back. Okay. Look at here, there's a clicker. How do it? Bend it. There's a click here, bend it. Okay, once you're doing this, you have to close it. Remember, these two caps, you have to hold on the hand. Do not put on the bench, because otherwise it's contaminated. Okay, what are this needle for? Some of the biochemistry reaction is fermentation, for example. There is no need oxygen. However, some of the biochemistry reaction need oxygen. So there are some of the wells right here, you have to block it. So you have to block it, block it, block it, block it, block it, block it, and break it, and then break it. Which means these reactions need oxygen, and some of them do not need oxygen. And then we'll give it to TA, we'll do the incubation for you. After incubation, you see the color change. The bottom line is that the whole classroom need to be the same color. If I see some of different color, which you're contaminated with something else. And we're not gonna read the code, okay? It's too complicated. Then you can take it home on Thursday. That will be your gift. If you don't like it, you can just give it to me, we'll throw it away. If you like it, put on your bench and take it to the home. If you want to keep it for a longer time, you have to freeze it. Put in a bag, freeze it. You will keep the rest of your life. If you don't like it, just put on the dorm, the bed, bench top for a couple of days. It will start to shrink because the solution will be dehydrated. If you want to keep the longer life, put in a freezer. Okay? Very good. That's a good try for you. So I have all these. I have a couple boxes I'm going to bring, and you guys can do it. That is one pencil for one student. Okay, everybody get a practice. And you will pick from your oxidase test E. coli side. After you pick doing that, please using oxidase reagent to do the confirmation test. Okay, so you need to arrange the time how to do it. So that's the first one, is entrance tube 2. Now, the remaining question is why we need to use this. This is use a multiple channel biochemistry test with 12 substrate and compare the color code to code book. And every color code represents one bacterial culture. Okay, now why we can do that? Because most of these tested bacteria belonging to a group called Enterobacteria ACA. They are all very similar characteristics, gram-negative, large, and facultative, non-endospore forming. And very important, lactose fermentation positive. And the gas fermentation positive. And in other words, pink colony from Macanthi agar. So, you cannot use the traditional methods to do the identification. It is hard. Therefore, we use EnterTube 2 compared to the code book. We will be easy to identify it. And one of the rapid testing methods. However, we will skip this code comparison. Okay, it's too complicated. And especially if you did not do a good job of the aseptic technique, it brings all the bacteria there. Then there's no way to compare. It's confusing. So I'm skip that side, but you get a practice how you use EnterTube. You will have fun. Okay. Second one, we will be doing some enzymatic testing. Enzymatic testing of bacteria. So what this means, bacteria has an enzyme, sometimes can use other energy resource other than glucose. So for example, they could use protein if they have protease. So how we do it? We will prepare a agar which is skim milk agar. So when we need a skim milk agar, I believe it's in the cooler. Skim milk. What do you mean skim milk? No fat, is that right? The whole milk, 8% fat. 2% fat reduced milk. And skim milk, which is no fat. So what else is dominant in the skim milk? Is protein. What type of the protein? Casein, is that right? That's casein. So we can test whether bacteria has protease. Could break down casein actually is we call it a caseinase. So how we do it? You have a skim milk agar, we'll split them onto two. On the left side, you will label Bacillus subtilis. On the right side, you will add E. coli. And then what we do, a similar gentle streak. Okay, by the way, this is not streak plating. This is just we make some smear there. Okay? So we do some gentle streak. And then we're going to do the incubation at 35 degrees Celsius for 24 hours. If you see one side, there is a transparent zone on Thursday, which means it is Proteus test positive. At this moment, we call it a caseins test positive. Okay, so that's the first one, skin mucus cargo. The second one, starch shock. Okay, bacteria could use glucose, but bacteria sometimes has to use starch. What is starch? Disaccharide. There is two major components. Do you still remember? Amylase and amylopectin. And if bacteria are going to use a starch, they must have amylase. So we want to test whether bacteria has amylase. So how we do it? We're using a starch agar, spread them onto two. On the left side, we're going to do the same thing, add Bacillus subtilis. On the right side, we're going to add E. coli. And then we will be doing 35 degrees Celsius, 24-hour incubation. Now the question is for you. How do you know the starch is used? How do you know the starch is not used? Some people say you see transparent zone. No. How we do it? How do we test the starch? Down, iodine. Is that right? Starch gonna react it with iodine. So if you see one side, the color is still purple, which means it's negative, because starch is still there. If you see the other side, there is no color change. Oh, sorry, all the color is changed. Then it is positive, because the starch is breaking down by amylase. So we need to do a confirmation test. Is dump iodine on Thursday. Okay, is that clear? Because when we grow the bacteria there is no way we know whether we use the starch or not. So how we do it? Dumping gram iodine. It's the same thing. If you see the purple color there, a brownish color, we won't say purple to brownish color, then we will say the starch is still there because it's reacted with iodine. So there's no amylase. If the amylase has been hydrolyzed to use the starch, then it's going to be the colorless because it's changed. Okay? So that's a little bit twisted from your mind. Okay, last one, what we're going to do. Some bacteria could use lipids. So we have an agar called sprit blue agar. And in this agar, we are going to do the same thing, but a little bit different. On the left side, you're going to streak with Staphylococcus aureus. On the right side, you streak with E. coli. And we will incubate 35 degrees Celsius, 24 hours, and we're going to see the transparent zone. If you see the transparent zone, which means it's positive, what this means? Which means bacteria use lipids, which means bacteria has an enzyme called a lipase, because they could be hydrolyzed lipids. Okay? All right, now something I want to say. We don't have Staphylococcus aureus. So we grow this in the lab, in my lab. There's 2 tubes. We don't just have this on the slump. So how we do it? You have to shake it before we do. Add a loop, pick some of the liquid, then you streak it. Very gentle. And a tiny picking. Otherwise, the whole agar will be like flooding. Okay, make sure, pick a little bit, and then you do a little bit circle, and it will be growing there. So, Staphylococcus aureus, you can pick from these 2 tubes. I put right here. And the others, we have Bacillus subtilis is right here. And also E. coli is right here. So, today gives you a job to do is to arrange your work how to do. We have a confirmation test. We have these agar tests. So what we do? Half of them do confirmation test first. Then half of you can do these 2 tests first. And then you find the time to do Staphylococcus aureus and E. coli, you split brew agar. Okay? Now, how do we recognize these agar? We're going to give you some of the examples. What is this agar? Split brew agar. We already said brew. That's brew. What is this guy? Skim milk agar. Looks like a milk there. Then what is this? Starch agar. Okay, if you don't know, what you do? The first thing first is a label. Make sure these outside are dried. Okay, label it if you don't know. Name, batch number, the bacteria we're gonna put it there. And don't forget, this inside, please dry a little bit also. Remember, the inside of the agar, please dry it. These has been there for a while. Okay? Otherwise, these condensated liquids, it's called capillary water, will damage the agar. You'll get bacteria everywhere. And after you do the inoculation, please just hold on like this a little bit, stay a little bit, then give it to the TA. Don't flip it over immediately, because this is liquid. Okay, this is liquid, remember? If you flip it, it goes everywhere. Then what you find, you're going to find a big line of Staphylococcus aureus on Thursday. There's no way to do test. The whole tube, the whole plate is positive. So these are some of the techniques you should know. This is gives you time to arrange your work. Okay? So let's repeat it, what you're going to do. 3 plates for enzymatic testing. Everybody got a beautiful pencil for inter-tube testing. That is lab work, label, give to TA to incubation. And don't forget, you do the confirmation test. Is oxidase test. And methyl red confirmation test. And Vogel's Prescale test. Reagent A and reagent B. You need to arrange to do very well. Okay, that is, and don't forget, oxidase test. You do that later on, because we need to do this intra-tube first. Otherwise, Yaga, you dump all the reagents, then you go, no way to figure out which colony is. So, do the intra-tube first, then that plate's put on the side, we can do oxidase test. Okay, that's what we're going to do.